Like Senegal said, we're really excited to have Dr. Sebastian Alvarado back with the CNI. Big words, but his research is really about understanding the molecular boundaries of biological plasticity, right? So again, I've been saying your brains don't grow, they're molded. Or really entire bodies. So while genetics provides a foundational framework for evolution, he emphasizes that it doesn't tell the whole story. His work explores how plastic molecular substrates enable genomes to dynamically respond to environmental changes. His lab primarily focused on African chitlets and other animals that are capable of actually altering their body coloration, such as Arctic mammals and cephalopods. So, despite the importance of plasticity in shaping traits and evolution, many of these reversible molecular processes, some of them are called DNA methylation, RNA editing, are still not fully understood. His research investigates how these adaptive mechanisms operate independent of essentially your genetic predisposition. So beyond the lab, Dr. Alvarado brings his expertise to the entertainment industry through Thwack, consults on science fiction projects, and writes books bridging science and storytelling. He's also an alum and mentor for the summer program in neuroscience excellence and success, furthering his commitment to fostering the next generation of scientists. Please, everyone, give a huge welcome for Dr. Sebastian Alvarado, who will lead us through our CNI Accessible Neuroscience lecture, Plasticity, How Experience Shapes Traits and Generations.
Thank you so much for the lovely introduction. It's a real pleasure to be here, as it always is. Can I share? Perfect. I'm gonna send a request. I'm gonna try and keep everything under the hour. I did add some stuff that I was really excited about that I'm like, oh, this is too much fun to leave out, you know? But can you guys see the slide? Great, fantastic. Okay. So I try my best to fit as much as I can. If I talk too much, I'm sorry. I'm happy if anybody wants to interrupt and ask a question saying you're going too fast or anything like that, please go ahead. I'm all ears. And if I don't see it in the chat, I'm hoping that Senegal will alert me to it so that way I don't miss out on any great questions. So I always like to start with a few comments on the history of biology. I think context is always important. I think all science, as objective as we try and be, is entirely, completely political. We won't talk about science today, but we will talk about science a few hundred years ago. Does anybody know who this guy is? I mean, I'm used to the classroom. Perhaps you do, perhaps you don't. This is Jean-Baptiste Lamarck. He came before Darwin, and he published this very interesting collection of manuscripts called the Philosophie zoologique. He's a French scientist who did some really interesting work. And when I was taught biology, I was taught about him in the context that he had this idea about how evolution worked, but he was wrong. In fact, it was explicitly told, we were given multiple choice questions, like which one of the following is correct about their assumptions and hypotheses about how animals evolve, and if the answer was which of the following is not the right answer was this guy. He had this idea of the inheritance of acquired traits. The famous example that's typically given is this giraffe elongation of the neck, that it was an acquired trait within the a generation of one animal's lifetime that they could then pass on to the next generation. So if, the first generation of giraffe could reach and stretch out just far enough, they would be able to eat, these branches up high in the tree, and then they would give birth to a progeny that would have a little bit longer neck. And then that animal would have progeny with an even longer neck, and so on and so forth. So the idea is you would acquire these traits within your lifetime, and then you would pass them to your next of kin. As a function of your lived experience. Now, this was heavily criticized, often for the wrong reasons too. This challenged a lot of ideas that were then presented by Charles Darwin, who put together this idea of natural selection, meaning the environment acts on biology, and then biology adapts or increases one trait within a population as a function of how the environment interacts with it. The difference here is that Lamarck presented this idea of agency, that the individual could change themselves and then affect that change in the next generation. And that could be a repetitive, iterative process. Let's say a conversation between the individual and the environment. In the other chaotic domain, it was the idea that Darwin put forward that everything is random. However, the way that the environment acts on the individual will shape what comes out of the wash in a way. This was a much more favorable point of view because of the church, simply put. The idea, sorry, I'll go back. The idea here is that agency, giving humans, or back then men, the agency to decide for themselves, took away the power the church had over people a few hundred years ago. And, some people might even argue today as well. But the idea here is that because of that political structure and the influence of the church, in fact, the idea that even tenure even exists in the first place is because of the church. I'm not picking on the church. I'm just giving you that context, right? That political backdrop, to the point where there were many people who were against Jean-Baptiste Lamarck. This is a very famous painting that was painted of him presenting his life work to Napoleon. And in addition to having someone like Napoleon at the time reject his work, it was depicted in this painting. He threw it to the side. He didn't think much of it. He had many detractors within his community, the big one being Georges Cuvier, who was just mean. He wrote this line that was then retracted from a eulogy that he wrote when he passed away. And it was perhaps Lamarck had lost the use of his own eyes through the principle of disuse, which was his own idea of use and disuse, through the way these giraffes elongate their necks. And in the later years of his life, he had gone blind. So think of your own colleagues at your workplace. When you pass away, they start cracking these jokes based off of the concepts and the ideas that you tried to build as a way of explaining why you went blind before you passed away. So this is not science, but you wouldn't be too surprised to find out it's still a little bit like that today, right? But there was this really important political drive, and it wasn't just Georges Cuvier, it was Augustus Weismann. There were lots of scientists that all collected works to disprove this, and often fueled by interest from the church and the governing party at the time. So this did some stuff, and I'm going to comment on that a little bit later. So I just kind of want to plant that seed now, and I'm going to rush you through some 100 years of molecular biology. In fact, the last 100 years of molecular biology. I'm going to go through this fast on purpose. I hope some of it sounds familiar. Perhaps you've already heard of it, but just to kind of catch up from where Jean-Baptiste Lamarck did his work to today. We all know about Mendelian genetics, the pea plants, this monk that spent all his time going through all these traits, crossing, and realizing that, well, there's something that's very interestingly being transmitted between generations, and it can be predicted with math. We can come up with mechanism. We can start understanding how breeding two different organisms can carry a dominant or recessive trait. And this turned into a formalized science, right? In the early 1900s, we were starting to realize that these genes can be mapped. We started understanding how it is encoded, what molecule it's encoded in, specifically DNA. And then we started to learn how to manipulate it. From this, we started developing new technologies to separate DNA, to read the alphabet that writes the letters of DNA, to understand how they code for protein. And essentially, there was this explosion of technology as we got into the late 1900s, the 1980s and 1990s. And we had this kind of age where we really embraced this world of genetics, that we assume that everything that causes anything comes from a gene, a mutation. We identified several genes that are responsible for disease, and that really built up our assumptions about how genetics work. We basically thought, well, there's some 10,000 to 100,000 human diseases, and we just found out that a gene can be responsible for disease. Therefore, the logic is that every gene can explain every single disease. Obviously, we don't say that now, but I remember growing up then, and we totally said that back then. And this led to an explosion. We wanted to sequence the human genome. We figured if we sequence the human genome, we can understand every single gene and how it contributes to every single human affliction. This work took up a good decade. Cost $1 billion and some 300 scientists working both in industry and academia. And more importantly, a lot of what we understand about molecular biology and genetics comes from these three model systems here: fruit flies, C. elegans, and yeast. Okay, don't forget that. There were mice, but mice take really long to breed. They're a lot harder to work with. When it came to actually building this rule set and understanding how these things work, it came down to things like E. coli, yeast, and fruit flies. And when it came to a lot of genetics, these three that I circled in red were kind of the workhorses of what we understand about molecular biology. So, but wait, can our genetics explain everything? Well, we wanted it to. However, I'll show you this piece of this paper that came out rather recently that took all of the insurance information in Aetna and familial relationships as a control for genetics, as well as looking at fraternal and monozygotic twins. The idea being, if you're looking at people who are genetically similar, is that a stronger predictor of disease through health insurance claims? Or is your area code a predictor of disease, in the sense that they actually had all this information, it was all centralized in one place, and we could use it to ask these questions. And we found out that like a 70% of human disease that are filed through these claims are often tied more to your postal code than they are tied to your familial connections. And this is mining fraternal twins, monozygotic twins, as well as familial connections. So clearly your genetics isn't explaining everything. And if you aren't convinced by that, I often like to use myself as an example. I did 23andMe some 3-4 years ago. I found out my ancestry, right? A very helpful tool in understanding that. But I also found out something else. I found out that I have the genetic muscle composition common in elite power athletes. Now, let me stand up because I feel that this doesn't really come through if you don't see this, right? And clearly there is a certain amount of environment and what I eat and how I exercise, things that I'm trying to change now in my life. I don't know if I'll be able to use this slide anymore. I hope I don't have to use it because I get in peak physical shape and I get to show off. But the idea is that like my environment is entirely what I eat, how I interact with people. All of these things play into it, and these are strong predictors of health outcomes, and we've known this for a really long time. But because we understood genetics and we understood that this is something we could, we could resolve disease down to the effects of these genes, we figured, okay, well, but let's forget about the environment. Let's just focus on what we understand. So this is where a lot of my work begins. This is where a lot of what I'm interested in. I'm interested in the epigenome, the epigenetics of how these things work, and epi just being above genetics. That's all it means. And often I like to consider it the dialogue that happens between your genes and your environment, right? The language that's spoken between these two things. It comes down to the cell, but specifically within that cell, if you were to take out the nucleus and the 37.2 trillion cells that make you up, we would find, if we slice it in half, a big bowl of spaghetti, right? That spaghetti representing DNA and protein all wrapped up. If you were to unwind one of these chromosome fibers, you would get this thing called a chromatin fiber. If you were to stretch it out even more, you would realize that all of your DNA is wrapped up in these little proteins that allow it to supercoil and pack itself in a really small space. These proteins can get modified, and that can determine how much they unravel or how much they tighten up, and that can determine gene function as well. And the DNA that wraps around these proteins themselves can also be modified with a chemical called methylation. It's basically the addition and the removal of a methyl group. Now, the simplest way that we're going to stick to today is this methylation is like an on or an off switch. If you see a lot of it in front of a gene, typically that gene is turned off. If you see very little of it, that gene is turned on. And what we've learned over the last 20, 30 years is that this modification is a strong indicator of how cells become different from one another. Because if you look at all of the cells in your body, they're probably genetically similar, because let's not forget, you were a single cell embryo, then you became 2, and that genetic information was copied exactly the same. Then you became 4, and that information was copied the same. And now you're 37.2 trillion cells with the same genome, but with cells that have completely different functions, cells that function in your eye, your mouth, your tongue, your muscles, etc. So there is this diversity of cell types that is heavily tied to how genes are turned off to make sure the right ones are on in the right tissues. We take it a little step further. We decide to look at how those changes affect animals in their wild and like in the wild and in their natural environment. I always like this example here because, on October 1st versus 26 days later, you have this Arctic fox that was photographed at a zoo. I believe it's Cleveland, Ohio, that this animal was photographed, and you can see that it's starting to get this white coat. This is really common in Arctic mammals, and it's going from this brown coat to this completely white coat. And all that's changing is really the length of the day as well as the temperature. But this is a really robust and dynamic, robust change that happens every single year. They get this white coat, they lose this white coat, and this happens over and over and over again. Now, we can't assume that they're genetically mutating, but the way genetic information is being used in response to the environment is creating this trait to manifest itself as well as to disappear. And that's what we're really interested in. So let me also elaborate. DNA methylation is heavily conserved, whether you're a plant, fungi, or an animal. Invertebrate mammals, it's really, really common. It's present with the machines that add it, as well as the ones that remove it, as well as where it's found. Now, here's a bit of a cruel joke that molecular biology tried to play on epigeneticists. These three model systems don't have DNA methylation. Let me rewind. Right here, we've got Saccharomyces cerevisiae. This is the yeast. No DNA methylation. We've got Drosophila melanogaster, fruit flies. This line right here, all the white filled circles indicate an absence of DNA methylation. And then, of course, C. elegans, the worm, an absence of DNA methylation. Let me go back a bit more and remind you that the fundamentals of molecular biology were all learned from these three animal model systems. Meaning we have this giant blind spot in our understanding of DNA methylation simply because of the way history has focused on genetics, which isn't to say genetics isn't important. It just becomes this pinhole view of what molecular biology is and how traits change constantly in response to their environment. So the big joke being, even though we want to study DNA methylation, we realize all of the animal models that have been used to understand it simply don't have DNA methylation, and that's just bad luck. Good luck for me now, but not so good luck for the people who had to try and figure it out, let's say in the 70s and the 80s. So what models should we be studying? A big philosophy that our group does here at Queen's College is that we study anything and everything that makes itself a good model. We're in a golden age of biology where you can adopt a lot of different tools to do some really fantastic things. And I don't think there isn't an animal model that I can bring into my lab, or I'm sure there's a few of them, but I try my best to work with everything that that could be a good tractable system. So what do we study? We study animals that are capable, that have a shared genome but are capable of change, and usually really interesting change just for us. If I were to do this in humans, the best example I could work with are monozygotic twins. Obviously, we know that monozygotic twins, they're genetically identical, and often one twin can develop a disease the other one cannot, and that can really only be attributed to these epigenetic effects. However, how many monozygotic twins can we find that are willing to donate the brain, that are willing to give tissue whenever I want, right? It's a bit more of an issue here. So we tried to pivot to other animal models. In the past, I've worked with a lot of ants, but right now at Queen's College, we work with a diverse set of animals. We work with a lot of fish, specifically African cichlids. These have a very interesting story I'm going to go into a bit further. We study African cichlids from Lake Tanganyika as well as Lake Malawi. We look at local bluegill. If any of you fish and you go out into the area and you go fishing, bluegill are really common stockfish you'll find all over North America. We look at a handful of Arctic mammals, Siberian hamsters, snowshoe hare, because of that color change I was talking to you about earlier. And we also do some work with a lot of invertebrates, a lot of cephalopods, as well as daphnids. There isn't enough time to go over all of these. I hope to one day in the future. But for the sake of simplicity, I'm just going to focus on this African cichlid here called the Tilapia burtoni, because they can change color. And they have really interesting social behavior. They can both change social rank and they can change color. And they do this within their own lifetime multiple times. And it's for that reason we figure, well, their genetics aren't changing, but their colors and their behaviors are changing as well. The story of cichlid fish, this is you'll see this in the chapters in many books about evolutionary biology. They comprise a family that evolved super quick. Some 2,000 species evolving across 3 lakes in about 10 million years. Well, 10 million years may seem like a long time to us. In terms of evolutionary timescales, this is really an eye blink. We know over the last 100 years of taxonomists and biologists visiting the lake that morphology and behavior have been hallmarks driving selection and speciation of these many different species. And then we sequenced them in 2014. We sequenced 6 focal species that make up most of these families. And we said, hey, they're not that genetically different. If anything, they're more like cousins. And I want to take a beat to ask you to appreciate the diversity you see in front of you. This guy right here, the one with like the big eyes, who's just kind of, this guy is the ancestral link to all of these fish here. And this is just how they look in terms of their morphology. They also behave and occupy different parts of the lake. Some of them go deep, some of them go only in shallow areas, some of them live in on only rocks, some of them live in these deep shell basins where other snails drop their shells and then they live within those shells. Others form these very interesting bowers. They have very diverse life histories and strategies and behaviors in addition to these different colors, and apparently they're all genetically very similar. This becomes a really interesting puzzle for someone like me to get involved in, which is one of the main reasons why I've adopted them as an animal model. We're specifically interested in their colors. Color is really important in these fish. They're very colorful fish. In fact, a lot of fish keepers use them in their show tanks because of their colors. Males specifically show these colors, and females choose the males, and they'll often use these colors to make those decisions. There's 2 main functions of nuptial coloration, so male-specific coloration, and one is to compete with other males to show, don't mess with me, right? I'm trouble, as well as to attract females. I'm going to specifically talk to you about Tilapia burtoni. It's native to Lake Tanganyika in East Africa. They have a 3 to 4 month reproductive cycle. All of the machinery that's missing in Drosophila, C. elegans, and yeast is present in this animal system. And what's really cool is that they actually mouth brood. So females will drop their eggs, males will release their sperm on top of those eggs, and then females will scoop them up into their mouth. And they'll hold on to them for about 10 days before they spit them all out. And this is a really helpful strategy because it gives us access to the single cell embryo very early on, and we can do all kinds of transgenic experiments, and we can manipulate genes and things like that. But most importantly, there are some robust behavioral phenotypes that relate to social status, to crowding, and reproductive potential. I'm going to quickly go over this work that was done by my postdoctoral advisor, Russ Fernald at Stanford. Essentially, what he worked with is he identified the social hierarchy of these fish. Specifically, you have dominant males. You can see they're brightly colored. They have this black eye bar underneath this eye, this red humoral blotch. And when you look inside their brains, you actually notice that a lot of the neurons that are important for reproductive behavior are relatively large. The receptor that this neuron releases is very heavily expressed, and they have these very large gonads. I've opened up many of these fish. You'll see that they're like those inflatable long balloons that you see at the kids' parties. Big long balloons, right? That make up the testes. Now, this dominant male, if you put him next to a larger dominant male, he turns into this individual here. He loses all of his color. These neurons that are responsible for reproductive behaviors shrink. The receptor that receives the hormone released by these neurons decreases its expression, and that balloon kind of deflates into this tiny, tiny, tiny gonad. What's interesting is that both of these guys can go back and forth multiple times over the course of their life. Their genetics aren't changing, but their social biology, the neural connections, the way that they behave, all these things are changing. And we can measure this in the same animal many times. So what about morphology? I mentioned their colors. Specifically, these are some of the experiments we've done in the lab, either playing with the downwelling light and the substrate that they live on. You can get these different colors. You can get blue fish, yellow fish. You can get something close to a blue fish on a green substrate. Also, playing with the intensity of light also seems to make a yellow fish when there's an intense light. And as you decrease the amount of light, these males end up becoming darker as well as bluer. And you'll notice that the males have this really large morphospace, the space in which they can change color between blue and yellow, whereas females don't really have that. But they have a lot of this lightening, darkening ability that males don't have. So they can get really, really, really dark, but they can't really turn blue as much. For the sake of simplicity, I'm just going to focus on the blue-yellow dualism that you typically see. They happen in nature. It happens in our fish facilities. It happens rather often. We did this really simple experiment to see, well, can we control this coloration in this fish? Can we change a fish from blue to yellow? And can we do it within the same individual? So we designed a very simple experiment where we took a fish, we moved it into a blue background. And then from this blue background, we took a piece of its tail before we moved it to a yellow background. And then we took a piece of its tail before we moved it back to a blue background. And we figured, first, let's see if they change color. And then after that, let's see if we can resolve it down to cells and specific changes in the epigenome. This was our first time point kept in blue gravel. This is our second time point kept on yellow gravel. And this is our third time point where we return them to blue gravel. Every row is the exact same fish. Now, about 100 years ago, if you see this blue fish here and this yellow fish in two parts of that lake, you think you're looking at two different species, because that's how we classified these, based off of very simple traits. They look similar, but these ones stay in one part, these ones stay in another part. Perhaps they're different species, but this is literally the exact same individual. So we figured, okay, we can control coloration in these animals. How can we resolve this to cells? The cells that make an animal have its color, at least these fish, they rely on what we call chromatophores. And essentially, these are cells that exist between two states. They're either aggregated, where they hide all their pigments, or they're dispersed, in which they spread all their pigments out so you can see them really brightly. They kind of work like this. This is a scale from a zebrafish, and you can kind of see them moving from this dispersed state, so these melanophores moving from a dispersed state into an aggregated state. And this is just from zebrafish. Zebrafish aren't very colorful, no offense to people who study those animals. Our fish, this is the least colorful part of the fish, and you can see that there's red, orange, yellow. There are these iridescent pigment cells as well, but there is a lot of diversity in that color. So we said, okay, let's look at these pigment cells and see how they change. We essentially found that yellow pigment cells are overexpressed, but they were also able to disperse their pigments much farther. So all of our yellow fish had more yellow pigment cells, and they dispersed that pigment farther, whereas the blue fish did not spread that pigment out at all. We did a candidate screen of many different genes involved in pigmentation, and we found ourselves narrowing down our interest on endothelins, because they're involved in a lot of different fish pigmentation pathways and processes. Very briefly, the endothelin ligand, we have it too. We use it to develop the early migration of our neurons while we're developing, but we also use it in our gut for smooth muscle contraction. But essentially, this is a ligand that binds a receptor here, the endothelin receptor. And when it does that, it changes intracellular phosphate, and this causes that cascade of pigment aggregation and dispersal. And we said, okay, if this gene is really important for pigmentation, perhaps it's changing as the animal goes between these yellow and blue conditions. We did find that the endothelin receptor, subtype B, was actually overexpressed in blue fish and underexpressed in yellow, meaning something was turning this gene off as it was becoming yellow, and something was turning this gene on as it was becoming blue. If you already forgot, DNA methylation, the thing that we study, acts like an off switch. So we made the assumption that perhaps DNA methylation within this gene is increasing as the animal is turning yellow, and it's decreasing as the animal is turning blue. Here's what the data look like. So these are the first 3 sites that can be methylated within the start site of this gene. So genes organized in a linear way. Here we have the endothelin B receptor gene, and these 3 sites that can be methylated. One is 125 base pairs from when the gene starts, 160 base pairs from when the gene starts, and 259 base pairs when the gene starts. And specifically, we found that one site had this increase in DNA methylation precisely when the expression went down. And if it's acting like an off switch, we identified this interesting relationship that might be regulating some of the coloration that we're seeing in our fish. It isn't enough to just see this. We figured, okay, well, let's give drugs that specifically target this pathway and see if it also affects, does it only affect the melanophores, the black pigment cells? Does it affect only the red ones? Does it affect the yellow ones? And very briefly, we did a handful of different drugs, but this is what a piece of this fin looks like in these fish. You'll see some of these pigment cells are aggregated, some of them are dispersed. But when you gave drugs that specifically target that pathway, only the yellow pigment cells ended up aggregating all their pigments. So we're like, okay, now we've been able to kind of connect this epigenetic change to some physiological change. So to kind of rush through this, we figured when the animal is yellow, it hypermethylates its gene to decrease the expression of the endothelin B receptor. That leads to very little of it being embedded in these yellow pigment cells. So any amount of endothelin that gets released into the circular system just doesn't have the same effect. On the other hand, when a blue fish comes along, or when the same fish becomes blue, this gene becomes demethylated. When it becomes demethylated, it acts like an on switch. A lot more of this gene gets produced, it gets expressed. A lot more of it gets embedded in these yellow pigment cells, which causes it to be hypersensitive to any amount of endothelins that get secreted in the circular system. So in a lot of ways, we figured, okay, this is a way of tuning color in these fish through an epigenetic mode of action. So what about behavior? We don't want to leave out any of the neuroscience that we're developing in our group as well. We also said, okay, well, blue fish, yellow fish, maybe they behave differently as well. We designed a very simple experiment to show all of this behavior. And what we did is we induced our blue fish or our yellow fish by putting them on either yellow or blue gravel, and we covered a tarp around the side. So we socially isolated them for about 1 month. We made them blue, we made them yellow. And then we put them in a focal tank where they had 3 females that they could interact with, but on their one side, they had a blue fish with their own community of 3 females, or on the other side, they had a yellow fish with females to interact with. So you could see if he was feeling aggressive or territorial, he would go to this glass divider and he would fight with the other males. So he had the choice, do I fight with a yellow male, do I fight with a blue male? In addition to that, he had the choice to interact with females. He could try and reproduce with them. And a lot of this is just kind of measuring what kind of behavior are we going to see. Remember, nuptial coloration drives both mate competition. So he has the opportunity to, it drives male competition. So he has the opportunity to interact with other males. But in addition to that, it also affects female choice. So we can see this interaction with females as well. So we put them in this focal tank and we watch them interact with all of the fish that we see here. And what we found differences. We found that our yellow males were a little bit more aggressive and our blue males spent more time entering the pot. We have this little terracotta pot that we put in our tank, and this is kind of like the territory that they claim. And it's a very important one because it's where courtship happens. It's where reproduction happens. So we basically saw that our blue fish were spending more time going into and out of the pot. Whether it's reproductively motivated, we weren't sure. After all, they might be hiding. They could be hiding, and they might have this aversive response to these males to their left and right that are aggressing them. But what we also found is that yellow males spent more time doing this threat display. It's called a lateral display. You can see a little video of it on the right here. And essentially, yellow males did a lot more of this threatening behavior to other yellow males than blue males did. And in general, we've seen a lot more of this threat type behavior. So we said, okay, behaviorally, they're starting to do different things, even though a blue fish can become yellow and a yellow fish can become blue. So we wanted to see if it in fact is motivated by reproduction, could we look at the sequence of behaviors? So what we did is we plotted these things called transition matrix models. And essentially they plot, they give you a qualitative view of the probability of one behavior to follow the other. Wherever you see a very thick arrow, there's a higher probability. Wherever you see a thin arrow or no arrow connecting these nodes, you see a very low probability of one behavior following the other. The size of these nodes represents the occurrence, how much it happens. So the larger the node, the more that behavior happens. And what we basically found is that our bluefish spent a lot more time doing reproductive behaviors consistently one after another than our yellowfish did. So we said, okay, maybe the entering the pot is in fact motivated by courtship. Maybe it is reproductively incentivized. So we're starting to see how this color difference plays into female mate choice. and how they choose their males when they're visiting these different territories and finding the prettiest male of them all. So we're starting to kind of scratch the surface. We then want to say, well, what about social behavior? Remember, I was showing you those dominant subordinate males. Perhaps there's an interaction with social behavior and the colors of our males as well. So we set up a very simple experiment. It's called a dyad. You put 2 males, 3 females, and essentially we did it either on a blue background or on a yellow background. If they were on a blue background, uh, so I'll show you what happened in terms of the effects of the environmental color on this, but what we found is that after a month, always one male becomes dominant, the other males become subordinate. So here we have the color patterning, the stereotype of a territorial male and a non-territorial male. And we basically observed them. After 1 month, do blue dyads behave differently than yellow dyads? Is there a difference that we can tease out within a social community that we have control over? Funny enough, we saw no differences. We basically saw that the interaction between a dominant male and a subordinate male on a blue background was pretty much similar to that of the same type of fish interacting on a yellow background. In terms of the chasing, the biting, all of these aggressive behaviors that territorial dominant males show, it was pretty much the same between the two. So we're like, oh, I guess social interactions trump um, uh, 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 these color initiated behaviors, these induced color type behaviors. So we said, okay, let's do one more experiment, but let's match up blues with yellows. So we designed a really simple competition assay. It's a dyadic contest. And essentially we took this dyad that, because we already set up these animals, they're already in a tank. We have a dominant blue male and a subordinate blue male, dominant yellow male and a subordinate yellow male. And we basically matched them up in fights. We paired the yellow male with the blue male on a blue background, and we paired the blue male with the yellow male on a yellow background. And we figured, is there an interaction between the environment, which is the visual environment that they're reared in, with their fighting, with their tactics, and how they interact with one another? This is the part where I love to hear from you guys. Feel free to use the chat because this is where I get to have fun. I'm looking at hosts and panelists. Who do you think is going to win and on what environment? The way I like to see it, if you ever remember this show from the early 2000s, Deadliest Warrior, where they would pit different historical soldiers against other. So they put like Genghis Khan against Vlad the Impaler, and they would say, who's going to win, right? So who do you think's going to win between the blue and the yellow? Yeah, I watch epic rap battles of history too, but it's pretty much analogous here. So given what you know, what I've shared with you already, who do you think is going to win? Do you think yellow males are going to win no matter what the environment is? Do you think blue males are going to win no matter what the environment is? Do you think that blue males are going to win more often on a blue background? Yellow males are going to win more often on a yellow background? What do you guys think? I'm actively looking at the chat right now. It is right. I don't know if it shows up on your screen, but I'm hoping somebody can share what they think. We can take bets. I won $20 off of Mark Halber at the ASRC here in New York with a bet that he lost, thinking that he thought he knew. I'm not asking for your money. I'm just saying, what do you guys think? Anybody want to unmute and ask? It's up to you guys. So Johan says, I think that the yellow male will win in the blue background. Really? Okay. Anybody else? Come on.
I'm thinking of the blue because it seemed like the blue was more dominant.
Okay, the blue might win because they're more dominant. So here's a spoiler
Not sure.
alert. Home colored, I like that. So Senegal says, what about the home color advantage? There might be one. I know the answer, obviously, but yellow dominates across both environments. Okay, Quinn says that yellow. So we got a diversity of ideas. So I'm going to give you one more piece of information. This has actually been done in the past. But what happens is that these males weren't made blue or yellow the way that I did in this experiment. In this experiment, we socially isolated them and we put them on blue and yellow backgrounds. So they didn't have the opportunity to interact with anybody else. And they were brought there at 3 to 4 months of age, just before they developed their colors. Every experiment previous to this, they've done 2 or 3 of them, where they have looked at blue versus yellow males, but they always take those blue and yellow males from brown gravel substrates. They don't control the background. They also handpick them. They say, oh, look, there's a blue in that tank, there's a yellow in that tank. They might be different ages. We're going to just size match them and put them in a tank to see how they behave. They don't control for the fact that maybe they're yellow because of a very complex social life history where they got beaten up by a bunch of yellow fish. And because of that, they know that the only way that they can be an aggressive, dominant territorial male is also by being yellow. They don't know that perhaps they were beaten up by a blue fish and they want to take the opposite color. Like there's a lot of nuance in their social life history that could be contributing from this, contributing to their outcomes. I'll tell you this much. In previous work, yellow males beat blue males every single time. Not every single time. They had a higher probability, like 80% of the matches that they were paired up in on brown gravel, they usually won. But again, we never controlled for the environment. So does anybody want to change their vote? What if they don't fight at all? Oh, I wish they didn't, but they're vicious bastards, these fish. They're really mean to each other. Anybody want to so there might be a home color advantage. Anybody want to change their vote? It's going to be 50-50. All right, so time for the data. Blue males won 11 out of the 12 bouts. So a bunch of you lost a bunch of money. One of you did, who actually said the blue males. So interestingly, when you control for all of these things, you actually see that the blue males consistently win. Now there's a bunch of pet ideas that we're developing from this. We think that in the previous experiments, a lot of them were actually housed in the same tank on brown gravel, and they were housed there between 4 to 30 days before the actual battle happened. Some of the work that we showed here is the fact that yellow males do more threat behaviors. They are showing, but they are not physically engaging. They're showing they're threatening, and that might have an effect on the mentality of a bluefish watching this really aggressive fish just waking up every morning and doing all these lateral displays. So we think that there's a bit of that. But in addition to that, if you take a spectrophotometer and you measure the spectra of brown gravel and the spectra of yellow gravel, it is pretty much the same signature, except one is less intense and the other one is more intense because it is more yellow shifted. But the idea is that both yellow and brown gravel, as far as the spectra are concerned, the quality of the light, it's very much the same kind of light. So all these experiments we've been doing up until now has given the home team advantage in a way to yellow males. Now, two things come out of this, depending on the background. Blue males fight differently. Yellow males lose, but blue males typically chase a lot more and they will claim territory sooner in a novel environment than if they're on the same environment they were on before, which is kind of interesting. So we're hoping to explore that further. So this is really asking the question, the weight of the environment on your genome. We're now exploring this. We're going to be resolving this. Just like I showed you earlier, we resolved it to pigment cells. We're in the process of resolving this to neurons within the brain and understanding which ones are responsible for social decision making. And we will then, of course, do the next part, which is measuring the DNA methylation of individual genes that are probably expressed or not expressed as a function of social behavior. All of this to go back to kind of the initial question that I wanted to pose is that, now is a perfectly good time to open up a lot of what Jean-Baptiste Lamarck kind of started. This philosophy of how things that can happen with your own environment, things that can happen in your own lived experience can shape your biology. And while we don't have a lot of information or clues as to how that crosses generations, there is evidence of it. We've seen it with epidemiological studies in individuals who have gone through famine. We are also seeing it now in Syrian refugees. Recent papers that's come out this year and last year have looked at the imprint of DNA methylation as a function of people who have gone through that. And I assure you, you're probably going to be seeing it years from now in all the other people that are marginalized and going through a series of lived experiences. And I think it's a really interesting way because it's not that we're taking away from genetics, we're not taking away from Darwinism or anything like that, but we're complementing it. And we're introducing a more inclusive approach to how these things change in our lifetime. And I think that's just really fascinating because unlike being born with genes that do or don't predispose you to certain health outcomes or disease, such as my power athlete musculature, right? You can control your environment. If you are having a bad day, you can turn it around, go out for a walk, right? The idea of genetics and heredity means that you were born that way, and unless you can change that gene in a subset of the 37.2 trillion genes that make you up, chances are you're probably not going to be able to change that because you're born that way, right? But you can change your environment. You can shape the dialogue that you have with your environment, and that in turn can affect your biology in interesting ways. And of course, I definitely think that has outcomes related to your health and disease as well, even if I'm looking at a fish here from East Africa, the East African Great Rift Lakes. Okay, so with this, I'd like to end with thanking my collaborators and my team, who I'm also grateful for. They're really amazing people. Dr. Fong, who led the study on pigmentation, Annalise, who's done a lot of the behavior on blues and yellows, Matt as well, who's doing a lot of the behavior on blues and yellows with Anastasia, Sarah Quisenio, who also joined my lab last year and is doing her master's project looking at how the colors of these fish changes in different environments. Fatima, whose data I showed you based off of the light intensity from light to dark. Andrew Claros, who did a lot of the modeling. Oh, sorry. A lot of the transition matrix modeling. Avi Kugadush, who, why does it keep doing this? I'm not touching anything. Avi, who's done a lot of the behavior as well. Don, who's part of my team. He's actually been working mainly on a lot of the squid work that we do, but I like putting everybody here. And then Yafim, who he is a chief lab technician from the physics department, and he's built literally everything that we use in our lab. He's got this great attitude in that he comes, he sees us trying to build and engineer things. He laughs at us, he makes fun of us, and then he walks away and comes back the next day with like the perfect solution to whatever we were trying to build. So we're very grateful for that. And of course, a special thank you to Dr. Maral Tajarian, colleague, collaborator, mother of my 4 children, and fellow faculty member here at Queens College, who's been with me as an incredible source of support throughout everything. As a scientist and a partner, and of course our funding partners, the Army Research Organization, NSF, CUNY, and Queens College. Happy to take any questions if you have any. Again, thank you again to the organizers of the CNI and for inviting me again. I really enjoy coming here, and I always love talking about my stuff. So
Yeah. Oh my God, that was amazing. Thank you. Big digital, I don't know, clapping, snap for Dr. Sebastian Alvarado. That was really interesting. I am a menace, so every time I I try to put together like what happens to summarize a talk, I always have like hot takes that, you know, we're in CNI, we can do it. We can have hot takes here because what I heard was scientists have ops. That's a thing. Poor Lamarck, a poor man. What I heard is that like if your parents or your grandparents say something to you, you can just be like, I'm gonna methylate your genes off me. I'll carry them, but I won't express them. So that's great. And I think, you know, but for real, like what we really understand is that there is some really sophisticated interplay between the environment and the individual, such that like you don't, you know, if I pick out a blue fish or a yellow fish, I don't know, I won't really know what it's capable of. I need to know so much about its environment to really understand like what those behaviors are going to be like. And I always say with our teachers that I think that they're, you know, I've been saying brain engineers, but I'm like, maybe I should just expand it as biological engineers because they change the environment for kids. And by doing so, you know, you guys are impacting these mechanisms that Dr. Alvarado, you know, works on, you know, maybe not the skin color or whatever, but but
oh, we
still,
all tan, right?
we do all tan. Yeah, are you going outside and playing? That's a very good point. So any questions from our lovely scholars? I could ask, but
I'm here for whatever you guys want to do.
yes.
Okay.
So Ray,
Hi. Sorry, I'm in the library, so I should be a little bit quiet. While I was listening, I was thinking about students' behaviors sometimes, and sometimes how we see parents reflected on the behavior of our students, and how us as teachers try to mold that in the classroom, teaching them a little bit of better behavior. So that's like the moving of the environment. Now, I was wondering how I can bring this into the classroom in student friendly kind of way. Is there something I can do?
There's lots of ways. I like the idea of exercise because I feel that sometimes just getting kids active and things like that, it's one of those things where you could really, like, it's something you could just, you know, that's something that that you can just work on right away, right? Like if you try and build muscle, you're like, oh, now I have muscle. Well, how did we get there? How did you build the muscle? If you want to go into the biology of it, or if you want to, if we're talking about behavior, we can talk about learning, right? Like every biological process that governs our being can be like, we can thank the geneticist who resolved it to genes, but now instead of just saying whether you have the genes or you don't, or whether you have the mutation or you don't, it's about how that information gets used, right? You make a bunch of decisions, and those decisions affect how you learn, right? If you decide to do your homework, if you decide to study every day, if you decide to, like, this builds into how you properly learn. If you cram, you're just not going to have the same outcomes. And like the studies have shown this over and over and over again. In fact, I love my field so much for like how stupid it can be, in that we've spent billions of dollars characterizing methylomes, DNA methylation, doing these transgenic experiments, and translating it to humans to tell you, eat well, exercise, surround yourself with, you know, nice people and a good social circle, and you'll be okay. Do you know what I mean? Like, you didn't need to give an epigeneticist that money to get to that outcome. And I remember my PhD advisor would always say this as a joke at the end of his talks, like, you know, thanks to all of our agencies, but at the end of the day, and after spending this much money, it turns out you should eat well, you should exercise. You know what I mean? There's nothing new there. So, but why not go through that process, right? If you're talking about learning, there's a lot of systems in place that govern that learning, that govern that social environment. I mean, these fish, just as an example, they are a product of their social environment, and we are all products of our social environment. And that lived history is something that we sometimes carry with us voluntarily. Sometimes we carry it with us involuntarily. It sometimes isn't our choice. And I think sometimes it's a place that, at least in the classroom, I've used to build empathy towards my students, knowing that I don't know what they've gone through. In fact, if the pandemic and going through that taught me anything, is just that our students are really, they suffer in very quiet ways that we're not going to know about. So that's one way that, and I often, you know, in the classes that I teach here, it might be too much to go into this type of detail at the secondary level, but like, I'll just be like, if you're listening to me, and I'll pick out the students that are and that aren't, and I'll be like, you know, John here listens to every single word I say, and James, I saw him sleeping. So let's imagine what's happening, right? You're processing this information, you're writing it out, so you're not only just internalizing, but you're then expressing it, right? That process means that the information that's coming out of my mouth, the words that I'm saying, is going through more circuits in this person's brain to the point that they can then write it out. And James here, who's been sleeping the entire time, I hope there's nobody here named James, but he's been sleeping. He didn't get any of that information. Both of them were sitting in the same classroom. Both of them were sitting in, you know, the same row. But right now, that information, the consolidation of that information has strengthened synapses. And effectively, this requires certain genes to be turned on within those neurons to consolidate and strengthen that circuit. So that way, when you recall this information, when you're either writing it down or you're studying for it later, this person's going to be able to learn more effectively. So I'm literally changing your brain in front of you, right? And I always use the example also of like a pizza, where like you can kind of initiate, you can start talking about food, people get hungry, they start salivating. It's like, this is the power of a teacher over their classroom, right? I can, it sounds crazy, like it sounds not crazy. It sounds like evil, like I can control your thoughts, but in a way we truly can. We can shape people's ideas if they're listening to us actively, right? So I often like to tie things to that when it comes to the neuroscience. And then if you do this enough times, these changes become very, very stable. Your ability to recall this information, to become an expert, either in the field or in the practice of a specific problem set or math problem, whatever, is a function of these biological processes governed by the same rules. I don't know if that helps, but that's kind of how I play with it sometimes with students.
That is so badass.
Yeah, you got my wheels to turning to thinking how I can incorporate this already. Because even like that making decisions part is very important in our students. Because when you were talking about the kids sleeping and the other one paying attention, that happens a lot in the classroom. So it's basically more on decisions like, what did you do last night that you didn't go to bed earlier? Like those decision makings of how you're going to improve in school if you're making bad decisions.
So one thing I didn't talk about that I just occurred to me now is so DNA methylation is often discussed as like this on and off switch, right? That's what I talked about today. But the other thing that it's really good at is that it's a really good fingerprint of lived experience. I have these slides from another talk that I did on aging, but the idea is that as a fingerprint, you collect methylation throughout your genome across your tissues. And in fact, we've been able to use that molecular fingerprint to guess a lot of things. Myself and Maral, my wife, we've done work looking at chronic pain, and we've identified signatures of chronic pain, different types of pain, as a function of the presence and absence of DNA methylation, socioeconomic status, and DNA methylation. Prenatal, this is something that happened actually in Quebec. I'm from Canada originally. In 97, we had this ice storm that basically froze over all the trees and power lines, and then it broke. So there were huge sectors of rural Ontario and Quebec that got cut off from electricity. People born during that time have different methylomes than people born on the same day that didn't go through that stressful natural disaster. So your environment is literally painting your genome in a way that if we were to read it, we could say, you live in a humid environment. You are going to die of this. You went to space. Literally, the Kelly twins, one went to space because they were an astronaut, the other one didn't. I'm sure their parents tell them all the time. They literally read the genome and they for this marked methylation as well as many other things, and they found that there was quite literally a signature of that lived experience. Now, where am I going with this? When you think of your students, every decision they make, let them feel that one for a second. Every single decision they make, whether it's good or bad, is going to have it's going to paint the genome. And this is something we can read afterwards. Now, the really exciting thing that I'm trying to get to is that this is being part of this emerging field called molecular horology, which is basically the use of molecular clocks. And we're using this to date people's ages. Now, there's your biological age and there is your actual age. Your actual age being how many times you've been around the sun, but your biological age being a measure of how you've lived your life and how these lived experiences have accrued to affect your biology. For example, if I'm the kind of person that wakes up every morning, jogs for an hour, gives everybody a high five, everybody loves me, I love everybody, I've got this great energy, I take care of the people around me, they take care of me, I eat well, I might be 40, but I might be registering at like 32 or 25. I like to think I register at 21. I've never checked, but I'm going to say 21, right? I'm a 40-year-old man. Other people who live very unhealthily, guess what? This clock advances their age. They might be 40, but they're going to register at 75. And this is really scary because people have done these studies. We've done this stuff, and we've seen that they register at a much more advanced age. And this, of course, brings them closer to death. Interestingly enough, they did the study. In fact, the group that I did my PhD with, they published a study, I think 2-3 years ago, looking at 60 to 70-year-old men. And they basically forced them to change their diet. They got them to do mindfulness-based practices. And they were able to, after 4 to 6 months of doing this, now these are 70 60 to 70-year-old men, these are going to be the most difficult people that you want to, they get to change their habits, right? And they were able to turn back the clock. I mean, the molecular clock. We don't really know what it means. It's just something we're reading. These are tea leaves. I want to remind everybody, these are tea leaves. We don't know how this functionally causes you to age, or so. We just know that this is kind of like where the dust settles. And as a function of just checking where that dust settles, well, we can say all the people who live like this, the dust settles like that. So it is really reading tea leaves, but really expensive tea leaves.
I feel like it's a bit more.
Huh?
It's a little bit more than tea leaves.
It's expensive tea leaves, right? But the idea here is that we're seeing these changes and we're associating them to different populations, and we're realizing like we can rewind the clock. They rewound the clock 4 years, I think it was. I have it here. Yeah, sorry, I don't know why I'm just, yeah, 2021, 43 males aged 50 to 72, an 8-week intervention that included a change to diet, sleep, exercise, relaxation guidance, supplemental probiotics and phytonutrients, and they looked at saliva DNA. And they used the molecular clock here, Horvath's clock. It's the one highlighted in blue that can be really robust, specifically in a handful of tissues. And they basically found that they were able to decrease the biological age by 2 to 3 years. So take with that, you know, what you think it might mean, but it seems that we can shape our biology in more than one way. So when you're talking about your students and what they should be doing and how they should be living, this isn't so much about neuroscience as it is about everything, right? Because all of your cells are governed by these same rules, or these same processes. There's more than one way in which you can be healthy, unhealthy. And that also translates to whether you're learning or you're not learning, or the idea that, oh, I can't learn it, it's too hard, I just don't get it. I'm like, well, maybe there's just, you have to take a different strategy to learning that would be more effective. But once you have it, you know, at the end of the day, it's all biology, right? It's all, we're all working with what we've got. If we're just aware that less of it is out of our control, and we start to understand what more of it is in our control, we can do more with our agency. See, I brought it back to the beginning of my last.
You did it. You did it. So Sebastian, you have to move us. We
Sure.
have a focus group so we can continue this conversation.
Absolutely.
Thank you so much for this talk. We're not done yet, so we're just going to move over to another link. And